These are the speakers from the cell biology lab, and they're going to tell you about their work studying possible DNA damage repair genes in the eukaryote Tetrahymena thermophila. And I'm going to let them go ahead and take it away.
Hi, good evening, everyone. I'm Taylor. I'm here with Connor and Melody, and today we're going to be talking about the Tetrahymena gene BAD-CP is not regulated during the DNA damage repair response.
So the DNA damage response is a series of reparative processes the cell has to dispose in reaction to some sort of DNA damaging agent. Examples of these DNA damaging agents can be seen above the DNA on the graphic, and examples of these DNA damage responses can be below the DNA on the graphic. But it's important to know this sort of process as one, this is a very important process to cell viability. It's important to keeping the cell DNA healthy and structured from the frequent bombardment of these DNA damaging agents that could otherwise disease the cell. And it's also important because this sort of response has been conserved through all organisms throughout evolution. So finding something in one organism can lead to finding that one thing in a multitude of other organisms. So the model organism that we use in this class is known as Tetrahymena thermophila, also known as T thermophila. And the reasons we use this organism is that it has a well-studied genome with genes known to be similar to those of human genes. And it's also known to be naturally very efficient at the DNA damage response, where it actually cuts up its own DNA during a section of its mating process known as conjugation. And in this slide, we are given two mRNA level profiles for two separate genes. The first profile on the left is for RAD51. This gene is known to be involved in the DNA damage response in almost all organisms. And it's to be noted that there are two peaks of upregulation during the conjugation phase, which is denoted by the C at the bottom of the graph. And this is to be expected, as we just mentioned on the slide before, the T thermophila cells cut up their DNA during conjugation. So RAD51 being a part of the DNA damage response would be expected to be upregulated during this point of time. Moving on to the graph on the right, this is the mRNA level profile for our gene BAD-CP, which currently has an unknown role, but it's to be noted that there are also two peaks of upregulation during conjugation. This could be denoted to a whole lot of different factors other than DNA damage, but in the context of this experiment, it's important to note this similarity. And given all this preliminary and background information, we were able to come up with the experimental question of whether our gene named BAD-CP played a role in the DNA damage response or not. And to figure this out, we use the processes of Pcr and Gel Electrophoresis. And to give a brief overview of both processes, Pcr involves using a double-stranded DNA template from a genome, and we then put gene-specific primers into the mix so that we are amplifying and annealing and then amplifying a certain section of that genome, and in our case, it's our gene. And this will then happen a multitude of times until we have millions to billions of copies of our gene. And we can then visualize this in a gel electrophoresis by aliquoting our PCR samples into wells. And because the DNA is a negatively charged molecule, it will move to the positive anode of the box when turned on, thus separating the DNA by length, the shortest DNA being able to move farther than the longest, and also by quantity. The brighter DNA bands having more DNA quantity than the dimmer ones. But to start this whole experiment off, we really had to design the perfect set of primers to amplify our section of DNA.
So like Connor had mentioned, we first began our experiment by finding primers that fit our gene-specific DNA or our BAD-CP. So we would have to find primers that would anneal to our gene-specific sequence in order to create enough copies to test expressions. So there was a certain criteria that had to be met in order for these primers to anneal and to pretty much maximize the annealing potential for our gene. So we picked one with no homodimers. And as you can see shown in blue, there is one intron with our gene. So we can expect that our bad CP or our gene-specific cDNA will be smaller than our gDNA when we do gel electrophoresis. Yeah, so using PCR to anneal primers in our gel electrophoresis to visualize our results did indicate that our primers do anneal to our bad CP or gene-specific DNA. We know that primers did anneal because we have expected bands at the gene-specific cDNA, which is present on our actual results on the right. We can, although we didn't get a band for our gene-specific gDNA, we can attribute that to some PCR error or some experimental error from our part. But we did know that the primer pairs did work because, again, we have presence with the cDNA, and we can confirm that there's no contamination of the reagents due to no bands in the H2O here on the on the right. So after primer validation, we had to do a reverse transcriptase PCR, so RT-PCR, followed by gel electrophoresis. We were used to measure DNA damage in treated versus untreated cells and then to induce DNA damage, cells were treated with the DNA damaging reagent, which is hydroxyurea. And then we had a lyse and had their RNA extracted and then converted into cDNA following the same PCR steps mentioned before. So as you can see, RAD51 is slightly upregulated in lane 2 as shown in green. And then same thing with our bad CP, which is our gene-specific DNA. Same thing, our lane 2 is supposed to be upregulated, and then our control group, which is RPPO, should have the same expression for both.
Yeah, so based on this, we can see that the bad CP gene expression actually does not increase after inducing DNA damage. Again, RAD51 and RPPO are our controls for this experiment. RAD51 is known to play a role, so we should expect to see an upregulation in its expression as shown in lane 2 by the brighter band in comparison to lane 1. RPPO is our loading control, so we use this just to make sure that we used equivalent amounts of cDNA in our experiment, and we can conclude that we did because we do have similar band intensities. In terms of bad CP, it was a little hard to visually decipher whether or not there was upregulation or downregulation, because you can see in lane 1 and lane 2 for bad CP, the band intensities seem to be a little similar, which is why we had to move quantitative analysis, which is shown on the right. So in bad CP, we don't really want to say that there was downregulation because the number 0.1x isn't really significant enough to say that it was downregulated. So we just say that gene expression was not increased. If it was increased after inducing DNA damage, we should expect to see expressions such as in RAD51 in the green bar, which you can see a dramatic upregulation in gene expression. So with this being said, again, bad CP is not directly involved in the DNA damage response. Although you do see 2 upregulations in the gene expression map, this does not imply direct DNA damage response involvement. So this lack of significant upregulation kind of moves us toward some type of indirect involvement after DNA damage has occurred. So with having this, again, if we truly did want to see if upregulation or downregulation truly occurred, we would need to do more experiments to truly validate whether or not downregulation occurred to a significant amount. But with the information that we do have, we can redirect research to bad CP possibly being involved in tumor suppression as a preventative response to DNA damage that could possibly be cancerous. There's very limited information in what we do know about bad CP, but what we do know is that it is an ARF-like 2 binding protein, as well as the homologs that shared similar gene expression to bad CP. And also we see this theme as a hypothetical protein, an ARF-like 2 binding protein. In the research that we had done outside of class, the ARF binding proteins, again, are still not well researched. But what we do know about the ARF family is that they are involved in tumor suppression, which is why we believe that bad CP can be used as a preventative response, as a tumor suppressor in cancerous DNA damage. Special thanks to Dr. Kristen Moore, Amber Antonucci, Harriet Lee, the Department of MCDB, as well as the University of Colorado Boulder. And then questions?
All right, and same thing, if you have questions, feel free to unmute, raise your hand, or put them into chat.
Do you think it might be possible to see differences in expression if you backed off a little bit on loading the amount of sample? Because some of the bands looked a little bit blown out, and I'm wondering if, and I'm not sure if this is possible, but do you think if you like in half the amount that you loaded that you'd be able to see differences in the band intensities?
No, that is a great suggestion though. We do know that if we were to really kind of, again, validate whether or not true downregulation occurred, we would definitely have to do more experiments. And in further experimentation, we could definitely kind of reduce the amount of cDNA that we use in our experiments to see whether or not this impacted band intensities.
The gels looked really good. Impressive gels.
Thank you.
I think we underestimate how hard it is to get a gel to work, right? And then you go and do real research and you're like, wait, what just happened? They're really, really hard. So you guys did a good job.
I think cell bio students would agree with you. All right, any other last questions for our presenters?
We got someone asking about showing the bands.
Which bands?
Maybe if you could go back to one of the slides where you showed the quantification of the intensities of the bands in the gel. Like this one, Will, is this what you're asking for?
Yeah, that's perfect. Thank you.
So it looks like there's a question that also came up in chat saying, what is the clinical application of this research?
So DNA damage is, again, it can contribute to disease such as cancer, which I guess is the most notable one in terms of our research. So it's really important to understand how these genes contribute to, again, whether they are directly repairing these genes or acting as a preventative response in response to like further damage that could be done to the cell after DNA damage. So I guess that's mainly in terms of bad CP, what the clinical response could be for this is just how does this respond to, like, how does it prevent or, you know, kind of respond to this DNA damage in order to prevent further cell cytotoxicity or even just cancer as a whole.
Yeah, and sort of to add on, there's some genes that are known to be involved in this response and other genes, a whole bunch of other genes that are not known to be involved in this response. So gaining a better understanding of what proteins and genes that are expressed during this can help us, further in the future, maybe manipulate them in order to prevent diseases like cancer.
All right, and one more. This is a question for your knowledge here. What type of organism is T. thermophila? Is it a bacteria or a prokaryote or a eukaryote?
Pretty sure T. thermophila is a eukaryote.
It's a microcilia organism. So if that makes sense.